Good morning once again. Here, as a neurosurgeon involved in the management of tumor-related epilepsy, I will try to answer to the question, how does epileptogenicity happen? That's not an easy question to solve in 10 minutes, and I will restrict the talk on the specific focus on glioma epileptogenicity and in what we call epileptocentric hypotheses that deal with change in neurotransmission and in electrolytes within the glioma. First, in fact, the glioma, to understand more this question, has to be envisioned as a rewiring disease. The glioma changes its peritumoral brain environment to its own advantage. And in fact, electrolytic changes we observe are required for glioma cells proliferation and migration. Glutamate acts as a growth factor for glioma cells, and then the glioma has to promote glutamate, and GABA acts as a regulative factor of migration and proliferation of glioma cells, so it has to be turned down. So as a consequence, we observed an increase in neuronal excitability due to the increase of glutamate within the extracellular space and due to the increase of potassium in the extracellular space. Concomitantly, we observe an altered neuronal inhibition because GABAergic signaling is changed, and in part due to a change in concentration of chloride within neurons. We will see that. First, regarding glutamate, we observed an increase of glutamate release within the extracellular space. It is released by glioma cells, it is released by activated microglia, and it is released from systemic blood vessels from the systemic circulation. In addition, in ADH-mutated gliomas, we observe an accumulation of 2-hydroxyglutamate within glioma cells that may act as a steric analog to glutamate. In addition to this increase of glutamate release within the extracellular space, we observe a lack of extracellular glutamate reuptake. There is a lack of glutamate reuptake by glioma cells, by non-tumoral glial cells, by microglia, and by neurons, because all of these cells lack the EAT transporters that normally reuptake glutamate due to glioma signaling. So the addition of an increase of glutamate release together with a decrease of glutamate reuptake induces huge increase of the glutamate concentration in the extracellular space, which has been shown in animal models and in humans. So it may produce neuronal hyperexcitability in the vicinity of glioma cells. And to date, the extracellular concentration of 2-hydroxyglutarate within humans is still unknown, to my knowledge. And as a consequence, the increased extracellular concentration of glutamate induces three main effects: cell death excitotoxicity, the neuronal hyperexcitability that produces seizures, and the tumor growth factor. Regarding hyperexcitability, which is the question of this topic, in our group we made preparation and we studied samples of human brain taken within and the peripheral parts of glioma, and we made electrophysiological analysis under physiological conditions, and we found that epileptic activities on this human sample, you can see spikes, were triggered by a glutamatergic signaling. In addition, we found that the same epileptic activities were triggered by GABA-A signaling too. It was quite astonishing, and such epileptic activities were triggered by GABAergic interneurons. And to understand better the underlying neuronal networks involved in such epileptic activities, we made unicellular recordings using sharp technology, and we found that GABAergic interneurons systematically fired before and at the beginning of such epileptic activities. And you can see in the histogram that they fired the line in green before pyramidal cells that are shown in blue or gray. So it is quite surprising because we have an epileptic activity which is triggered by a GABAergic signaling, and we observe a paradoxical response of pyramidal cells to this glutamatergic signaling, because one should expect a hyperpolarization of a pyramidal cell following a GABAergic triggering. But we observe in more than 60% of cases, as you can see on the left side in blue, that 60% of pyramidal cells were paradoxically depolarized following a GABAergic inhibition. So it is the demonstration of an impaired GABAergic signaling within neurons in the peritumoral environment, and it is due to an impaired neuronal chloride concentration. And this depolarizing GABAergic effect were linked with the density of glioma cell infiltration and with the grade of malignancy. So to understand better the underlying mechanisms, we studied the possible mechanism of this altered chloride accumulation within neuronals in the periglioma environment. And we show an impairment of the chloride cotransporter within neurons. We show a huge increase of NKCC1 within the neuron's membrane. NKCC1 is a chloride loader that accumulates chloride within neurons, and we show a decrease of KCC2 expression, and the KCC2 is a chloride extruder through the cell. So the increase of chloride accumulator together with a decrease of chloride extruder induces a huge increase of chloride accumulation within neuronal cells, which explain why, following GABAergic signaling, we have a depolarization of pyramidal cells and not a hyperpolarization. So these abnormalities were triggered by glioma cell signaling. It is not perfectly understood for NKCC1, but some glioma cells signaling are involved, and it is more known for KCC2 signaling because it is under the SRC tyrosine kinase pathway. So, the take-home message, in fact, we know from clinical practice that glioma evolution and epileptic evolution are linked together. And to understand this quite simply, you can envision the glioma as a raw wire-wing disease. It uses glutamates as a growth factors. It decreases GABA because it is an anti-proliferative factor, and it induces hydroelectrolytic changes to its own advantage. And in fact, all of this point structure, which is the peritumoral cortex, which is the infiltrated brain within the early periphery of glioma cell, which is a front line for the wide war against glioma, because it contains isolated glioma cells that account for the oncological prognosis. It contains the epileptogenic fosses that account for the epileptic prognosis, and it contains plasticity. So, in fact, because glioma and seizures progress with time, and because the epileptological and oncological prognosis are linked with the extent of resection, and because glioma cells extend beyond MRI defined abnormality, here had advocate an early and maximal functional based resection for both oncological and epileptological prognosis, in fact linking toward a functional neurological surgery. I thank you for your attention.